Hi everyone, and welcome to today's Biology Daily Booster. This is number 10 in our Paper 2 series, and it is advanced information listed topics for both foundation biology and higher tier biology. We're going to start off on Unit 6 today. Now, the first bit of Unit 6 is looking at different sort of sampling techniques when we're talking about going out doing sort of ecological sampling. Before we can delve into that, what we need to know are four kind of key terms. First one, distribution, which just talks about where species are found over the total area on where they occur. Population, number of organisms of a species in a given area. Relationship is interaction between species living in the same area. And sampling is what we do when we count a small number of the total population so that we can study it. What we need to understand then are the different techniques that we can use. So even though this isn't listed as one of the PAGs, it's still potentially going to come up as a question anyway. So let's go through the basic bits of equipment and how we use them. Quadrats, first of all, that's this thing here. You probably went outside with a bit of plastic or a bit of metal. Sometimes it's literally just a square, sometimes it's got grid actually structure inside it as well. The quadrat has a fixed area, usually ½ meter square, and what we're going to do is we'll use the quadrat, place it in a certain location to be determined based on what we're sampling, and then you count the plants within it. Now, in terms of how we use a quadrat, we can use completely random sampling, or we could use more of a systematic system, which we'll look at a little bit later on in this booster. Pooters, this thing at the bottom here. A pooter is a way of collecting little invertebrates. So what we've got, one straw with no mesh on the end. The other straw does have mesh on the end, very important. The one with mesh on the end is the one you suck. So you basically place the straw without the mesh on the end next to your little bug, suck through the other one, and the bug ends up in your jar. Obviously make sure that you don't randomly switch which straw you're sucking through halfway through the experiment, otherwise mouthful of bugs if you're sucking through one without the mesh on the end. Third one, pitfall traps, that's this one here. We dig a little hole, stick a jar in it, and then we cover it with a little roof so it doesn't fill with water and drown the poor little creatures. This is what we're going to use to catch little ground crawlers. So beetles, for example, will be wandering along, fall down into our little pitfall trap and be there for us to count the next morning. Next up is the sweep net. We can see this fine figure of a man modelling for us. What we can see there is you go out, collect things like grasshoppers. It's basically a big net that you sweep through long grass. You catch the little creatures in the net and then we can count them. Transect lines are next. Now the transect line, quite simply, is where you're going to use a tape measure and lay it out. So if we've got the sea down here, for example, and we want to see how the distribution of plants changes over the beach, you lay your tape measure out across the beach, going from the sea further up, and then every meter you would place your little quadrat down. You can then sample what's present in there and carry on all the way up the beach. So it just gives us a way of working out what's present using that transect line. You could use it going out from under a tree if you were looking at light intensity, etc. Kick sampling, that's where you're going to go into a river and, as the name suggests, kick the edge a little bit, disturbs anything that's there so you can then catch it in a net downstream. And tree beating, stretch a big white sheet under a tree, give it a little bit of a shake on the branches, and then all the little bugs fall down on the white sheet, and again you can see what's present. It's all good and fine collecting all these things. We need to be able to identify them. And to do that, we use a thing called a key. Two types, spider keys or branch keys, and numbered keys. So this is our spider key. Start off, we have a plant in front of us. Then we're going to have a look at the plant. If it has broad leaves, we go to the left. If it has narrow leaves, to the right. If for the sake of this, our plant has broad leaves, and then we look at the edge of the leaf. Is it ragged or is it smooth? In this case, it's got a smooth edge, therefore it is a daisy. Easy. The numbered keys, pretty similar. It's just like one of those choose your own adventure books you might have read as a kid. So we've got our plant still, and we're going to look at it, and this time it's got narrow leaves. Okay, so we read, and it says go to question 2. So here is question 2. Then have a look at the flowers on it. They're bell-like flowers. We have a bluebell. That's as simple as using those keys are. They can give you a range of pictures of random little creatures and a key and ask you to identify a certain one of them. Just work your way through it nice and simply, look at the features that are there, and then eventually you'll end up with the name. One of their favourite things to ask you about is capture-recapture technique. When we're looking at how we carry out capture-recapture, then as the name suggests, you're going to go out, capture some, and then at a later stage, recapture them. What we do then is you go out on a particular date and you are going to then use a sampling technique to capture your little sample size. You count how many you've caught and you're going to mark them in a way that's not going to affect their survival. You then release them and then you come back at a later stage, so you decide how long you're going to leave it, and then you use the exact same method to do a second sample. What we then do is you count your organisms that you've caught, and you're going to be counting 2 things. First of all, the total number you caught and how many were marked. Once we've got that bit of information, we can work out the total population size. So what we do is your number in the first sample times the total number in the second sample divided by the number that were marked in that second sample. By doing that, we get an estimate of the population size. There are a couple of big assumptions we are making here. We are assuming that nothing died between our 2 sample times and nothing of that particular species we're looking at either immigrated or emigrated. So nothing moved into the area, nothing left the area we were sampling. Big assumptions, hence why it's only an estimate, not a true count. As we mentioned, there are a couple of different ways we can sample. We can use random sampling or a more systematic approach. So random sampling, we usually do this if you wanted to get an estimate of population of different plants on a field, for example. Then what we do is we use 2 tape measures and you lay them at 90 degrees to each other, as you can see in the picture. That gives you basically a grid. You can use a random number generator then to give you a series of coordinates, and then what you're going to do is go out and place your quadrat at those random number generated coordinate places. You then count the plants in there, do it however many times that you did, multiply up to the total size of the field. Now, the whole purpose of doing this using a random number generator is it removes any sense of bias. Because I guarantee that if you did this experiment where you went out with the quadrats on your school field and you were told to get an estimate of the population size of daisies, then after you placed your quadrat randomly a few times, you were kind of sitting there, oh, it's boring, there's no daisies. And then you started looking for them, and then conveniently the quadrat fell on that area. When you do that, there's bias. You're going to get a false size of the population. Because if you're deliberately placing the quadrat where you can definitely see there are daisies, it's going to overinflate that population size. Second option, the non-random sampling. This is when we actually want to study something. So if we wanted to see how distribution of populations change over a certain distance, so whether it be how the population of different plants changes as you move from the shade under a tree into the bright sunlight, is it that we're looking at how the populations change as we move away from the sea and further up a beach, like the picture down there? If that's what we're looking at, we need to use non-random sampling, because we're actually studying how it changes based on a certain factor. So this one uses the transect line, and that means we're going to run our tape measure away from whatever it is we're sampling, and then you place your quadrat at fixed intervals along the line, counting what is present in each one. From that, we can then obviously look at the distribution with those different factors, and then we can make our conclusions based on that. As always, if there are bits you'd like to go over in a bit more detail, you can use the video on the main channel, you can use your revision guide and your classwork notes. Don't forget to have a little look at some of the past exam papers, just to get a bit of a feel for the kind of questions that come up here, And of course, join us again tomorrow for our next Daily Booster.